Hello everyone, I am Dr. Sirajul Faizi. I welcome you in my YouTube channel. Today in this video, I am going to talk about estimations of protein by Biurate methods. So, in this experiment, our aim is to estimate the protein using Biurate method. This is very important methods, and as a rules of safety, I always suggest you cannot perform any experiments alone in the laboratory. Always you perform experiments or any other activity in the chemistry lab in presence of your supervisor, your friends. You cannot do anything in the laboratory alone. So let us start learning of today's experiments, and the experiment name is to estimate the protein using Biurate methods. You know what is the principle of Biurate method? Here the CONH, all organic peoples are very known about these functional groups, and this functional group is known as peptide, CONH, peptide. You know it is present in protein. We are calling polypeptides. So in polypeptide chain react with the copper sulfate in alkaline medium to give a purple color which can be measured at 540 nanometers. So reagent required Biurate reagent. Dissolve 3 gram of copper sulfate. How much? 3 gram of copper sulfate. And 9 gram of sodium potassium tartrate. 3 gram of copper sulfate and 3 times means 9 gram of sodium potassium tartrate. Just remember 1:3. 3 gram of copper sulfate and 9 gram of sodium potassium tartrate. Process in 500 milliliter of 0.2 mole per liter sodium hydroxide. Add 5 gram of potassium iodide and make up to 1 liter with 0.2 mole per liter sodium hydroxide. So protein standard 5 milligram BSA per ml. Bovine serum albumin 5 milligram BSA per ml. Now apparatus and glasswares required: test tube, pipettes, calorimeters. What is the procedure? Pipette out 0.0 means nothing, 0.2, 0.4, 0.6, 0.8 and 1 ml of working standard into the series of labeled test tubes. So here the important things, you have to label your test tube clearly and you have to write blank 0.2, 0.4, 0.6, 0.8 and whatever. Pipette out 1 ml of given sample in another test tube. Make up the volume to 1 ml in all test tubes. A tube with 1 ml of distilled water serves as the blank. Now add 3 milliliter of Biurate reagents. Then add 3 ml of Biurate reagent to all test tube, including the test tube labeled blank and unknown. Mix the content of the tube by vortexing or shaking the tube and warm at 37 degrees Celsius for 10 minutes. Now cool the content to room temperature and record the absorbance at 540 nanometers against blank. Then plot the standard curve by taking concentration of protein along x-axis and absorbance at 540 nanometer along y-axis. Then from this standard curve, calculate the concentration of protein in the given sample. So these are the 8 steps. You can follow these all steps carefully and do your experiment step by steps. Don't miss forget any steps, because each step have their own importance. And before performing this experiment, you should wash your glassware very clean. If it is not clean, then you will get the ambiguous results. So your all glassware should be clean. Your sample should be prepared carefully. Whatever the measurement I have given in this video, you can just follow the procedures. Then you will get your results, and whatever the unknown sample present, you have to report in milligram protein per ml. And this is BSA, is bovine serum albumin. So you can take another protein also, but this is the chemical is easily available in all chemistry and biology labs. So friends, if you like this video, then subscribe my channel, and thanks for watching.